On November 25th, 2018, history was changed when biophysicist He Jianqie announced on his YouTube channel that he had successfully impregnated a woman with embryos that had been edited using CRISPR technology.
Two beautiful little Chinese girls named Lulu and Lala came crying into the world as healthy as any other babies a few weeks ago. We're also sending a little bit of protein and instruction for a gene surgery. When Lulu and Lala was just a single cell, this surgery removed the doorway through which HIV entered to infect people. After birth, we again deep sequencing Lulu and Lala's whole genome. This verified the gene surgery worked safely. No gene was changed except the one to prevent HIV infection.
Jianqie used CRISPR to disable the CCR5 gene, a gene that encodes the production of a protein that allows HIV to enter a cell and replicate within the human body. The news was announced just one day before the Second International Summit on Human Genome Editing, sparking debate, discourse, and outrage among the scientific community. His experiment was largely criticized because he edited the twin cells when they were just embryos, ensuring that every one of the twin cells were now changed, including the gametes, meaning that their edited genomes could be passed on to their children, despite the fact that experts can't be sure what the long-term effects of such lasting modifications might be. The successful birth of these fraternal twins represents a significant leap in the use of genome editing, but scientists are still concerned about the long-term effects of procedures such as this. It's important to note that He Jianqi's work was never published. It was rejected by all the medical journals it was submitted to. A reporter from the MIT Technology Review got a hold of his manuscript and showed it to scientists, who were shocked to find that his claims weren't supported by his results, the baby's parents could have been under pressure to agree to join the experiment, and the researchers went ahead with the experiment without fully understanding what they were doing. These kind of actions from rogue scientists are cause for real concern. Before we continue, let's take a few steps back. What is CRISPR?
A stunning and controversial breakthrough in science has arrived: gene editing.
This is a milestone that could one day erase hereditary conditions.
Some fear the technology could be used to create designer babies.
This technology has the potential to change our DNA and the DNA of all organisms alive and extinct.
Could that lead to eugenics? Could that lead to new divides in humanity? I don't know.
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR sequences were first discovered by a Japanese scientist back in 1987 in E. coli bacteria. It's since developed into a powerful tool that helps scientists edit DNA with precision. It works by using an enzyme called Cas9 to cut DNA at specific locations using a guiding piece of RNA, allowing scientists to add, delete, or replace genetic material. This has the potential to revolutionize medicine by allowing scientists to correct genetic mutations that cause disease. It's also been used in agriculture to increase crop yields, and in research to study gene function, but the focus of this video is just going to be its human applications. That brings us to today, or at least a month ago, to the third International Summit on Human Genome Editing. Many scientists believe that there have been more CRISPR babies born since the He Jianqi controversy back in 2018, but perhaps have been dissuaded to publish their results after his 4-year imprisonment after publicly discussing his experiments. That's all to say that research hasn't stopped because of this controversy. Just the opposite. Scientists all over the world are constantly discovering new side effects and risks with CRISPR gene editing in embryos, as well as developing even greater breakthroughs. One of the biggest concerns with the use of CRISPR technology is the potential for shuffling large segments of chromosomes or causing other unwanted genetic changes. There's not enough evidence to convince scientists that there aren't significant risks to the offspring's life, or potentially their children's lives after going through gene editing at the embryonic stage. Scientists still don't know what the implications and risks are of cutting both strands of the DNA double helix, which is a necessary step in many forms of genome editing in embryos. Although there's been extensive research into the editing of adult somatic cells to treat diseases, as well as the embryos of mice, we don't know if there will be different consequences should those same changes be made at an early developmental stage. Like I said earlier, CRISPR works in embryos by cutting both strands of the DNA double helix. After the cut, the cells in the DNA try to repair themselves, but this method is typically filled with errors that can cause unwanted mutations. Nucleotides can add and delete themselves through this process, which can dramatically change the amino acids and proteins that it produces. Research has also shown that CRISPR-Cas9 genome editing can result in the loss of large portions of chromosomes, some of which can't even be detected using our current technology. Scientists worry that human embryos can't stand that kind of intervention so early in the developmental process, with one study showing that 40% of embryos couldn't manage to repair the broken DNA, and 1/3 of the embryos that could repair the DNA resulted in the loss or gain of large pieces of chromosomes in some cells. The big story from the summit wasn't even the research made to further progress the development of human genome editing, but the ethical concerns revolving around making these edits to embryos in the first place. Laws and regulations revolving around the type of gene editing that He Jiankui did have been established and even bolstered in China, but really haven't even been considered in the United States. Should the technology be made available, there's currently nothing in our legislation that prevents parents from ordering designer babies, in which they can choose the sex, eye color, hair color, skin tone, and even maximize the IQ level of their child.
Oh, no, it's disgusting.
This kind of treatment would likely only be made available to the wealthiest prospective parents, furthering financial inequality between the haves and have-nots. Skeptics and critics fear that CRISPR technology could bring a second rise to eugenics, which is the practice of selective breeding in order to guarantee desirable traits within one's offspring, which was used by the Nazi regime to justify their treatment of the Jewish population, disabled people, and minorities. Jennifer Doudna, one of the inventors of CRISPR-Cas9, even has worries about what her technology can do. Here, she's describing a dream she had, which highlights her fear of her technology being used for eugenics.
And one of the things that sort of brought that to the forefront of my mind was a dream that I had fairly early on, in which I was, I walked into a room and a colleague of mine said to me, Jennifer, I'd like you to explain the CRISPR technology to a friend. And he brought me into a room, and a person was sitting with their back to me. And as they turned around, I realized with sort of a horror that it was Hitler, and it was actually Hitler with a sort of a pig nose, and it almost looked like a chimeric pig-human sort of creature.
Scientists need to consider that the edits they make to this child's genome will not only affect their lives, but the lives of hundreds of people to follow. Critics will also argue that couples who are heterozygous for a life-threatening disease, or are not guaranteed to pass it down to their child, don't have to use CRISPR, but can turn to in vitro fertilization, or IVF. IVF allows scientists to create multiple embryos and screen each one for the potential disease. The doctor will then insert the embryo without the disease-causing gene into the uterus, which would allow parents to raise a healthy child without risk. Supporters of embryonic gene editing argue that it's critical for parents who are guaranteed to pass down a life-threatening disease, such as cystic fibrosis, sickle cell anemia, or Duchenne muscle dystrophy. These parents deserve a chance to raise healthy children as well, and the development of technologies like CRISPR will be critical for their ability to do so. Scientists and researchers also expressed their concern for economic inequity when it comes to gene editing procedures. In November of 2022, the FDA approved a gene therapy treatment for hemophilia B, which costs $3.5 million. In developed nations, this can largely be paid for with health insurance, but for those uninsured in the US, and for those living in low to middle income countries who don't have the financial infrastructure to support this kind of treatment, it's effectively useless. This also isn't the only one. There are more than 200 people who have been treated with experimental CRISPR tech, and nearly all of them are wealthy individuals who can afford the procedures. CRISPR remains in the early experimental stage, which means that no federal approvals are granted, which means that access is limited only to participation in clinical studies. Natasha Salome Lima, a psychologist and bioethicist at the University of Buenos Aires, highlighted that although low and middle income countries account for 70% of global cancer cases, a striking 2/3 of gene therapy cancer trials happen in wealthy nations. Doctors fear that exorbitant price tags of gene therapy treatment will prevent the majority of people who need them from being able to access them. Scientists also brought up concerns of racial inequity with use of CRISPR. There's a huge gap in DNA information for people of non-European descent, meaning that there are people from many parts of the world for whom these gene editing therapies may not work at all. This opens up the door for discrimination in treatment not only in countries abroad, but in minority groups within the United States, because genetics knows no borders. I know I've spent most of this video talking about the negative effects and consequences and unforeseen circumstances involving gene editing technology, but I want to finish up this video by talking about the many positives to come out of this summit meeting. First, I want to talk about Victoria Gray, a 37-year-old mother from Forest, Mississippi, who suffered from sickle cell anemia. Back in 2019, Gray was the first patient to undergo a new experimental therapy that extracted the stem cells from her bone marrow, corrected them with CRISPR, and reinserted them back into her bone marrow.
I think it is a great thing. Who doesn't want a good quality of life where you don't have to suffer? Because my disease I didn't choose. I was just born having to endure pain and hospital stays. It was a life that I felt wasn't worth living.
Her presence at the conference was to underscore the scientific community's shift away from inheritable DNA changes to somatic corrections. Then there was Alyssa, a 13-year-old from the UK who was diagnosed with leukemia, which is basically a blood cancer. Her leukemia affected her T cells, which compromised her immune system. Normally, leukemia in children is treated with either chemotherapy or a bone marrow transplant, but Alyssa's cancer resisted both.
Alyssa was only 12 years old when she was diagnosed with a form of leukemia that's 96% curable. She soon found out she was in the other 4%. Chemotherapy didn't work, leaving doctors to suggest a revolutionary new treatment called base editing. 6 months later, Alyssa is cancer-free.
I'm feeling happy, actually.
And why are you feeling happy today?
Because I have had new cells put into me.
Doctors took donor T cells and genetically modified them to seek out and destroy the cancerous T cells and replace them with the healthy ones. After more than 10 months since the treatment, Alyssa's cancer has remained undetectable. Gene therapists were able to modify the donor T cells by using a process called base editing, which can change individual nucleotides from one letter to another in the genome sequence. Scientists first reported that base editing techniques did not cause off-target DNA mutations on their experiments on rhesus monkeys, but did cause unwanted mutations in the RNA during embryonic testing. This unprecedented level of precision in gene editing within human somatic cells had not been achieved prior to Alyssa's case, and her treatment has opened the door for numerous children to overcome this form of cancer in the future. The world of gene editing, particularly CRISPR technology, is a double-edged sword, presenting groundbreaking opportunities for scientific discovery, as well as providing many social, ethical, and safety concerns. The controversial announcement by He Zhuanghui in 2018 brought these concerns to the forefront of scientific discourse, leading to an ongoing debate on the merits and dangers of genome editing in human embryos. The Third International Summit on Human Genome Editing highlighted not only the technological advancements in the field, but also the ethical dilemmas and potential for economic and racial inequality regarding access to gene therapies. While inspiring stories such as Victoria's and Alyssa's demonstrate the incredible potential of CRISPR to revolutionize medicine, the scientific community must continue to navigate the complex challenges associated with the technology. Striking a balance between innovation and ethical responsibility is essential in ensuring that CRISPR is used to benefit humanity without causing unintended harms or perpetuating disparities.